Back to my list of points about developmental disorder. The damage is done really early before you are born. That's an assumption. Does it hold up? Is it supported by evidence? Well, there's a growing body of evidence that autism is not necessarily prenatal. We now know that the brain, on average, gets larger after birth, which I'll show you, that there's tissue changes, inflammation, cellular changes, which I'll show you. There have been, what I don't have slides on, is that there are a series of animal studies with animals, mice, getting prenatal infections, and postnatally they exhibit behaviors that are consistent with autism, although an animal model can't fully produce autism. But the question of whether this is sufficient to cause autism or necessary or just a strong contributor, we just don't know. Whether there are other things that are necessary. This is the graphs of some of the graphs from some of the research on increase in brain head size. You know, when you take your baby to a well baby visit and they put the head circumference around and they mark that on a chart, and it's supposed to follow a certain trajectory, and in autism it jumps up a number of those levels in the first 2 years of life, not for everybody, but for a substantial number. And this was observed by a few people, Janet Lanehart and some others early on. Eric Kershane published a paper here. It went from 30th to a much higher percentile, up to 2 standard deviations in the first 2 years. By now, there are dozens of papers about this. Here's another one showing increases. On average, most people with autism have brains that are above average in size, which is different than the general population distribution. Most people with large brains don't have autism, so large brains don't mean you have autism. It's more that there's something about autism that gives a tendency to large brains. And that enlargement is concentrated more in the white matter, the part of the brain. There's the cells that live in the gray matter, and then these cables that go between the cells in areas where there aren't so many neurons that are wrapped in a white fatty material called myelin, and that makes the white matter, and I'll show you some pictures of that. This is a picture, the line, the straight line in the middle, the 0, is the control line, and comparison of head size in autism to controls starts out small, huge rapid increase in the first 2 years of life, so that's this line squeezed into a much shorter space. And then that increase stops, and by late teens to adulthood, the brain size can even be smaller on average. So there's a dynamic process going on in the brain, and it has a different trajectory than in typicals. One of the contributors to the idea that autism was a prenatal disorder was my colleague Margaret Bowman, who talked about changes in the cellular structure in certain parts of the brain consistent with developmental changes that would have occurred because those parts of the brain developed around 30 weeks in gestation. That was taken as evidence that this was prenatal, and I saw some years back I've seen grants get rejected for studying postnatal metabolism on the basis of it's irrelevant because the brain changes happen before birth. Dr. Bowman published data and now has commented on it. Here you see children and adults. So the top row is children, the bottom row is adults. The left is autism and the right is control. So in the children, the autistic sample from postmortem brain tissue, the cells are big and fat and plump, and in the controls they're smaller and paler. In the adults, it's the other way around, paler and smaller, bigger and plumper. And Dr. Bowman and her long-term colleague, Dr. Kemper, said that this suggests that there is some chronic, persistent cellular process going on during the life course, which they did not try to speculate on what it was because you can't possibly say from this kind of data, but you can say that there is something going on, and it's not just prenatal. This was a paper which I have found absolutely pivotal in my own thinking, and many people in the biomedical approach to autism also do. It was a paper with first author Vargas, senior author Carlos Pardo, a neuropathologist and neurologist at Johns Hopkins University, who was brought into this field by Andrew Zimmerman, whom I've mentioned as the pediatric neurologist who edited the book I mentioned, Autism, Current Theories and Evidence. Pardo identified in brain tissue from 11 individuals who died after a diagnosis of autism that there were cellular changes consistent with immune activation, microglial activation, astroglial activation, cells changing their conformation and what they do and their cellular markers in relation to immune stimulus. There's innate immune activation and adaptive. This was only innate, which is another discussion. This was a different finding than what people had found in the past. You cannot really see innate immune activation on an MRI scan. People had looked for these big scars, such as what you see in multiple sclerosis. That's more adaptive immune. That's what not what you see here. To say that there's immune change in these brains is to say something about the tissue of the brains that people hadn't said before. Interestingly, in the initial sample, the individuals whose brain tissue was donated died ages ranging 5 years to 44 years, and the intensity of that inflammation was the same throughout. After the paper was published, Dr. Eric Courchain gave Carlos Pardo a brain of a 3-year-old who was rigorously diagnosed with autism before death, and that individual had much, much more inflammation. And it makes you wonder if this rapid increase was an inflammatory process, and many of us are wondering that. Unfortunately, there are no direct ways. I'm going the wrong way. Oops, sorry. Let me fix this. There's no direct way of doing brain imaging of inflammation right now, so you have to figure it out indirectly. By now, the sample has grown substantially at Hopkins, although I don't believe they've published their larger sample. There was an individual a few years ago that Dr. Zimmerman told us about who had signs of brain inflammation, who had Asperger's. So it isn't necessarily associated only with severity. This is my own brain imaging research. What we did in our lab, which I'm only going to give you a little bit about now, was say, okay, if the brains are bigger, what parts of them are bigger? And first we identified that this area containing both yellow and white, that's the white matter, was bigger. Then we went back and divided up that area, and we found that it wasn't the whole white matter. It's this outer part. Where's the cursor? There it is. The yellow part, which was the part that was bigger. As it turns out, that's the part that gets that myelin sheath around the axons, the latest in development, after birth, toward the end of the first year into the second year, and really late in the prefrontal areas. And we found that prefrontal white matter was 36% bigger in our sample than in controls. That's huge for a brain. And sometimes I see little children with autism with big foreheads, and it makes me wonder whether there's some relationship there. We can't prove it. What Dr. Pardo did was, when my paper came out, his paper was already in press. He went back to his samples, and he measured inflammatory markers in the gray matter, the outer white matter, and the deep white matter, and found that the outer white matter had inflammation and the deep white matter did not. And he invited me to Hopkins. He and Andy Zimmerman invited me, and I thought I was going there to give a talk, but I think his real agenda was to take me in the lab and show me this. And it was, I think, probably the most moving experience in my own scientific career to see that my work had inspired something as profound as this kind of a connection. But it suggests that the brain, tells us something about what might be happening to the brain that makes it bigger. Now, why is the brain getting inflamed? There are many genetic diseases where brain tissue gets inflamed. There are also a myriad number of environmental factors that can contribute to brain inflammation. Oh, and by the way, I just forgot to mention that Dr. Pardo also identified oxidative stress in brain tissues, which is something that you'll be hearing about from some of the other speakers during the day. It's a problem of the metabolism not being able to keep up with the stresses on it. So Lilian Calderon in Mexico City studied people with children who had no history of neurodevelopmental delay disorder or whatever, who died abruptly from accidents, and compared the children in Mexico City and young adults with children and young adults in Minnesota, where in an area where there was not air pollution, and found that the children and young adults in Minnesota had neuroinflammation, altered innate immune response, disrupted blood-brain barrier, ultrafine particulate deposition, so the little particles that are airborne from your catalytic converter that cross your lung membranes and your brain membranes between your nose and your brain and many other places, because your body can't screen them out, and accumulation of amyloid beta 42 and alpha-synuclein, which are associated with Alzheimer's or Parkinson's in kids. This neuroinflammation has very similar cellular features and cytokines. Cytokines are immune chemicals to autism. And I brought Dr. Calderon to the Autism Society to speak about this last summer. It was her first exposure to autism, and she found this to be an uncanny similarity. Air pollution has already been linked to autism in several studies. So it is not out of nowhere that people are talking about air pollution. And I want to say that even the Autism Genetic Resource Exchange got an $8 million grant, and Clara Lajonchère was courageous, who runs that that entity within Autism Speaks, was foresighted enough to include an air pollution component in a genetic study, which is an example of linking environment with genetics. This is air pollution over China. This is a huge hunk of China, Shanghai, Hong Kong, and air pollution that's going to blow over the Pacific Ocean in the direction of the United States and Canada. I was showing you glial cells that were activated in the brain. There are glial cells in your gut. The body does not read medical school textbooks. The body does not know that you have to keep the brain in a different chapter from the gut. The body uses the same genes, the same molecules. They turn different ones on and off. But there's no intrinsic barrier that's fully non-permeable. And in fact, there's a whole nervous system in your gut called the enteric nervous system. And as a matter of fact, that nervous system doesn't only have neurons, it has glial cells. Glial cells have been like the handmaiden of neurons, but actually they're really important. And they've been called mind glue and the cells of the 21st century. It turns out that the glial cells, and Joe Coyle has written a lot about that, the glial cells in the gut perform immune signaling and barrier functions just like they do in the brain. And this article states that even though we don't know that much about these cells, it's predictable that enteric glia, gut cells, glials in the gut, are involved in the idiopathogenesis, the development of various pathological processes in the gut, particularly those with neuroinflammatory or neurodegenerative components. So this business of the gut-brain connection may have many bases, and one of them may be the cellular impacts on cells that are in more than one part of the body. This is something which I've just happened to put here. When you see news stories about genes being associated with autism, many times you see, oh, this gene causes autism. I think that it's fair to say that if you have an association, even in a small number, that gene may contribute. But because we don't know whether it's sufficient or necessary, if it's not sufficient, only necessary, or only contributory, cause is a bit of a strong word. So I just want to caution you on sometimes the exuberance that you see in the newspapers is a little overstated. So I'm going to point number 3. The brain is fundamentally and irretrievably differently structured and broken. Here's a statement from a scientist who had a paper out on a gene that affected both brain and gut. He told the reporter, you can treat the gut if you want, but that won't affect the autism because the autism is caused by structural changes in the brain. And I think that many of us have believed this or thought that this was what we're learning when we study the brain, the actual top-of-the-line cause of what we're seeing. And I've done a lot of brain work and brain writing, and I've come to the point of view that that actually does not make sense. So I'm going to explain to you why. So I've already shown you some of the structural changes in the brain. We have the large brain, some regional differences, the amygdala, various other regions, white matter differences. They're all associated with autism. But do they cause the autism, or are they caused by the same things that cause the autism? Now, I'm going to say this a few times in different ways so you can try and get it. But what I want to say is we've been so busy assuming that it causes the autism that we haven't asked what causes those problems. We assume it's genes. Do we know it's genes? No, we haven't. We don't really know. Could the structural changes in the brain be effects of chronic functional abnormalities, and could the chronic functional abnormalities be further upstream than the structural problems in the brain? So I talked to you about this brain inflammation and brain enlargement. I already talked to you that there is, for inflammation, a lot of reason to suspect that environmental factors can contribute to it. So something is causing these structural changes. Here is a paper, and this is complicated, so I'm going to walk you through it slowly. First, let me go back, to this picture here of my own work. The idea that this outer part of the brain that I have in yellow would be bigger. What people assumed was that it was normal tissue, only bigger, that the brains were bigger because there were more normal cells, more normal neurons in the gray matter, the stuff out here which isn't colored in, the gray matter, more fibers in the white matter. And then what they did, without evidence, because the scans that we had available to date didn't tell you anything about cells and didn't tell you anything about fibers, so it was an assumption. What they did was say, oh, the greater amount of fibers between the little areas of the brain out there explains how autistic people are so good at detail. And the lower number of tracts in the deeper parts of the brain explains why they aren't good at putting the big picture together. And this was assumed to be because of structural changes in the brain, except that nobody ever proved that that was what was really going on in the brain tissue. All they had, when you say, and from a scan, that it's bigger, it's just that there's an area and it looks white and it's bigger. You can't tell what that area is made of. So in this method, people are able to look at signals related to the fiber tracts of the brain, the cables, the white matter, and you can divide it up by what it is that you look at how the water is traveling along the tracts. If there was nothing there but water and the water could flow wherever it wanted, but if there are tracts, like if you have a bowl of spaghetti and you pour tomato sauce on it, the tomato sauce doesn't go through the spaghetti. It's restricted by where the spaghetti is. Similarly, in the brain, if there's water, it tracks in the white matter, it will track along the fibers, and it will only move freely in areas where there are not fibers. So you would predict that if there are more fibers, the water flow should be more restricted. So if there's no restriction, you call it isotropic. Isotropy. Iso is the same, and tropy means the tendency to go in any direction. Anisotropy means it isn't equal in all directions. So the more restriction there is, the more integrity of the white matter there is. So you would predict, if there's more fibers, that there would be more restriction. Looking at this outer part of the white matter, in this study by Sundaram and from Diane Chugani's lab, they found, no, there was less restriction. Not only that, there was more diffusion. So it's like there was more water in there and less structure constraining the water, which is the opposite of what you would predict if it was a normal brain with lots more cells. Here was an earlier paper from Western Ontario. The area of yellow, the areas of yellow were areas which had increased something called T2 relaxation. And what you need to know about that is it means that the water properties were different, probably more water rather than more fibers. It does not support more fibers, consistent with what I just said from the last one. Here's a picture, here's a slide with no pictures because I couldn't find any to illustrate it. They had the same kind of finding. The white matter had more water more fluid, less restriction, and the more it was like that, the worse the diagnostic score was, the more severe the autism was. The authors were very surprised. They had predicted the opposite. And they said that this might be consistent with neuroinflammation. They also pointed out that there was less NAA, which I didn't talk to you about yet. That's in the next slide, which I should reverse those slides. But NAA is N-acetyl aspartate. It's a substance that is found in neurons. You can measure it with an MRI scanner using spectroscopic techniques. And it indicates either how many neurons there are, how healthy they are, and interestingly, it's made in the mitochondria. In the mitochondria, so there are issues about mitochondria in autism. Well, in almost all the studies where they were able to measure any difference in NAA, it was less, suggesting that there's less density of metabolites. So even in the gray matter, rather than having more tightly packed cells, it's looser and more dense, suggesting a problem with either cell health or cell density. I would suggest it's probably more cell health for two reasons. Number one is the brains don't really get a lot smaller. And number two, there's a really interesting study, 4 papers actually in epilepsy, where if you have a seizure focus deep in your brain and a lot of seizures from there, that piece of the brain will have very much reduced NAA, and it will excite the same region on the other side of the brain that also will show reduced NAA. And people thought that was permanent damage. But in 4 different papers in the literature, this is not an autism thing, but with intractable seizures, you can go in surgically and take out the seizure focus. And then the irritant is gone, and the other side of the brain is not irritated anymore, and it gets its NAA back. It recovers in the brain. And this is something that's really profound, because we've been told that if it's brain damage, it's brain damage, and here is something where it's brain severe irritation, but not cell death, and something which can bounce back. So I think that's really important to understand here. So what I'm saying to you is, rather than structure causing function, if anything, function would cause structure, but really, function can impact structure, and then when you have the structure involved, it can impact function. And wherever you get into this loop, it's a problem, but you can get into it because of biochemistry or immune things, and not just because some gene messed up your brain. Here's a really, really big anomaly. Now, many of you may know the book, The Structure of Scientific Revolutions by Thomas Kuhn, and he talks about everybody has this mindset, and then these anomalies build up, things that don't fit the model. Like Galileo got into all kinds of trouble because he said the sun doesn't revolve around the earth revolves around the sun. It makes more sense, and they gave him a lot of trouble, but obviously he was right. It's just that not in his lifetime did he really get appreciated for it. But here we have a big anomaly. A lot of what I've been saying is anomaly, but this one is really interesting. Parents were telling Dr. Zimmerman and some other people that their kids would do better with fever. How many of you have kids who do better with fever? Yeah, a fair number. And they decided, let's test this prospectively. Here's the thermometer, here's a behavioral rating scale, what's happening. And they found that the majority of the kids actually improved in core features of autism in a way that couldn't be explained away just because they were tired or worn out from the illness. This is not consistent with static encephalopathy. If it was static, the hardwiring would not allow you to get better like that, even for a little while. So this has provoked an interesting amount of reflection and papers about what could cause this. Is it the environment impact on glial gap junctions? And this is something that Derek McFabe knows a lot about. Cytokines, membrane lipids, dysfunctional electrophysiological oscillations. The chairman of the neurology department at Albert Einstein has gotten into this, writing a paper on autism epigenetics and the locus ceruleus, which is a part of the brain that regulates state through noradrenaline. And he said it's dysregulated early and that transiently gets re-regulated in fever. It's a theory. These are all theories. But it really knocks your socks off to think that people can get better even for a little while. There are actually more and more stories and some documentation of improvement. There was a paper in children with bad diarrhea and regressive autism being given oral vancomycin to treat clostridial bugs that were abnormal variants, doing better while they were on the antibiotic and then getting worse again when the antibiotic was discontinued. Children who go on Clear's fluids for colonoscopy, starting to interact and even talk for a little while, and when you reintroduce the foods, it goes away. I heard a story from a very prominent researcher here in Canada that a 33-year-old low-functioning autistic woman came out of orthopedic surgery and into the recovery room and started to talk with her mother and told her mother, "I love you, and I am so sorry. I've been such a burden, and I know what it's been like, and I haven't been able to talk or reach out to you." And the mother and the daughter spent 6 hours in complete communion with each other, and then the woman went to sleep and woke up, and she was back in her nonverbal state. What does this tell you about the brain? Is that a hardwired disorder, or is that a chronic encephalopathy? It's a very challenging thing. In my group, we think that there was our guess is the anesthesia affected something about the electrophysiological oscillations in the brain transiently. Some children can talk briefly. They come up with these complete sentences during times of stress when they normally don't talk. Some children who have recovered a lot will start talking to their parents about things that were going on when they were considered to be autistic and completely out of it, that actually stunned and almost terrified the parents because they didn't think that the person was really taking stuff in. So, and there's also people who improve in core features of autism when they're on anti-epileptic drugs. So all these things are, which is a support for an electrophysiological brainwave kind of impact. These should all raise questions in the mind. Do you see what you believe, or do you believe what you see? Is the so-called brain damage, which I'm suggesting may not even be damage the way we're thinking about it, the cause of all the problems? We have functional problems in the brain. Are they caused by the large-scale problems, or are they caused by cell metabolism problems? What I want to focus on here is that the cellular problems that you'll hear about and that are being addressed or attempted to being addressed by biomedical approaches can affect all kinds of things in the cell, the energy, the neurotransmitters, the lipids, the glial cells that affect the way the nerves can function and the way they can network. These are cellular, widespread. They affect timing, signal intensity, and coordination. And they could explain a lot of the findings that are going on in brain research. So what I'm suggesting is that autism could be a dynamic, a different model, that it could be a dynamic, active consequence of challenges to cellular function throughout the body, including the brain, that these cellular changes, which I hope you hear more about in other people's talks, may be related to environmental influences, that altered cellular responses could be at the root of brain and body problems, and that this could explain the dynamic features, because the cellular status can change, even on a day-to-day or hour-to-hour basis, and many cellular problems can be treated. I'm going to skip some of the stuff on the brain because I don't have enough. Well, actually, I have these clocks aren't the same. Which clock is right? That one. This one's fast. Okay. So one of the things that we see in the brain in autism is that this is a typically developing person. Works better when I do it here. Where if one area is activated, the other area is activated in very close synchrony, and you can see these two lines are practically on top of each other. But in the autistic person, there's a lot of discrepancy. And that discrepancy is reflected in a lower level. This one has a functional connectivity, 0.79, and this one, I can't even read it, but 0.3 something, 0.31. So you see less functional connectivity, less coordination among the brain regions. In EEG, you can have sometimes less, sometimes more, depending on the power band. But the point is, let me show you this slide. This is from our own lab. So what we have here is EEG, and let me just walk you through. It's 5 to 8-year-olds, 9 to 11-year-olds, less complex stimulus, more complex stimulus. So the less complex stimulus is you sit there and you want to give the child something to do so they don't completely get distracted. So we give them the screensaver. You know the dots that come at you, those stars from the PCs. Put it on moderately slow. And then the complex stimulus is we put together selected clips from Fantasia. So it's very engaging, it's very beautiful, but a lot of complexity. And In the and so it's a simpler, predictable complexity versus something that's really novel. What the pictures, the lines are the areas of the brain where there was differences in the coordination between 2 points. Each line connects 2 points between autism and typical comparison groups. So in the screensaver, in the 5 to 8 years old, there was a modest, mostly reduction in the coordination between different brain regions with the screensaver. But when they looked at Fantasia, they got slammed, and they really couldn't keep up with the brain coordination that was being marshaled in the typical developing children. By far, most of this was a reduction in synchronization and coordination. By the age of 9 to 11, similar autism with similar severity, the individuals did not show that they could keep up at the same level with the typicals for the easier stimulus. But the more complex stimulus, they still had some decrement and also some increase in stimulus. So you see that this is a moving target. It's changing with time. There's some catch-up and adaptive function. What we're saying is that some of this is related to what we need to test is this being driven by the white matter, or is it being driven by other more dynamical factors? This is a very important concept that I want you to pay careful attention to because it's something that comes up a lot in autism literature. It's this concept of an excitation to inhibition ratio. It's not just in autism. People talk about it a lot in neuroscience. John Rubinstein and Mike Merzenich proposed that a lot of features of autism could be explained by an increase in the excitation-inhibition ratio. So here's a cartoon of the ratio. In the numerator, you could have too much excitation. In the denominator, you could have not enough inhibition. There are many things that can cause those changes both on the top and on the bottom, but if either one happens, the net result can be overall more excitation, which can make you more irritable, can give you hypersensitivity, can give you overload. This can contribute to many features of autism, such as the epilepsy, the sensory hypersensitivity, the sleep problems, and it can be something that occurs in other cell systems of your body. Inflammation and oxidative stress are excitotoxic, and they can increase the excitation-inhibition ratio. Huge numbers of chemicals can increase this ratio, and treatments can modulate this ratio. So this is a very nice concept, what you're looking for, that it's a very simple thing that can like a little backbone that holds a lot of things together. So finally, is there nothing you can really do about this? This is a child who had a symptom. She was a relatively moderately severe functioning girl with autism who could talk but did not communicate. She was nonverbal. She was a patient of someone in our clinic who noted that she was going around going, I'm okay, I'm okay. The girl had never had seizures, but just in case this was a manifestation of seizures, she got an EEG, and the EEG did not show, it was an overnight EEG. The girl had these episodes, there was no seizure activity. She turned out to have reflux, this was reflux. But even though she had no seizure activity, she had epileptiform spikes in her brain, and so much so that as much as nearly half of her brain was offline due to hypersynchrony around the spikes. But it didn't meet criteria for 75%, so the neurologist, the initial neurologist, epileptologist, who saw her did not treat her, but one of my colleagues who actually did her epilepsy fellowship here at Sick Kids did treat her, and she did improve cognitively and in language. And actually now there's some effort to get research funds to study whether treating these non-epileptiform spikes might be of some use. And actually all day today there's going to be a seizure forum here, so it's really getting to be a very important area. This method of looking at this is not in standard use clinically because it uses computer techniques which are not, they haven't been incorporated into standard neurology. And I think we need to think about that in terms of developing methods that are more sensitive to the brain electrical problems people may have with autism even when they don't have seizures. In terms of things not being able to change, these are 3 mouse models, Fragile X, Rett syndrome, and tuberous sclerosis. These are all conditions that are not autism but are frequently accompanied by autism, and they're considered to be developmental disorders with all the baggage that I told you, irreversible, genetically determined brain damage. In all of these 3 mouse models, the symptomatology was significantly reversed by a targeted molecular intervention. This really shakes up the thinking about what it is that's going on with these conditions, even when there's a known genetic cause, which there was in all of these 3 cases. And here are a couple of papers on Alzheimer's, a drug that inhibits something called tumor necrosis factor alpha, which I'll talk to you about in the next slide more. It's an inflammatory cytokine, an immune chemical that promotes inflammation. By injecting a drug that inhibits this into, it's not something you can do every day, but these individuals experienced rapid, although transient, improvement in cognition and in language. If that's the case, then all of the plaques and tangles in the brain and the volume loss and all, however much they may be contributing, they're being augmented by the chemical impact on brain function of inflammatory substances. Now, tumor necrosis alpha is something in the brain, something that in your system that can be triggered by bacterial or other exposures. So if you inject something called lipopolysaccharide, LPS, which is something that's in bacterial cell walls, when the immune system sees it, it thinks, infection, I've got to do something. And one of the things it does is it makes tumor necrosis alpha. In the bloodstream, in this mouse, in this rat model, it went up, the tumor necrosis alpha stayed up for 9 hours, in the liver for a week, and in the brain for 10 months. So what this means is that, if you think about it, it could mean that inflammation in the brain could be triggered, but it could be so persistent that if you just keep triggering it, it's never going to back off, and it will look like a static encephalopathy. But maybe it's just a chronic, persistent, currently re-triggered through some immune stimulus, chronic encephalopathy. Okay. I think I'm going to skip the next 4 slides because they're just reviewing some of the model stuff. And I believe people have the slides in their materials. Is that correct? Or you don't? In the CD. Okay, so now I'll just go through them really quickly. I was just going to say that the gene-brain behavior model, it's because you think it's inevitable, you look for drug targets, you're not in a big hurry, and I've already said all of this. I just want to get on to the rest of the talk. Whereas if you look at a more whole systems model, you look for strengths, preventable features, and look at this as a public health crisis. So here are some further features that I think need to be better developed. I've already said all this. There is not that much research going into the chronic features of autism. There is not that much research really thinking about active ongoing contributors. Even the people who are doing research on inflammation and oxidative stress talk about it as if its impact is on brain development. Now, maybe it is in brain development, but does it stop there? There's no reason to think that it stops at the level of influencing brain development. It seems to be something that causes problems every single day. Looking at brain problems as dynamic, most brain researchers trying to identify what's wrong with the brain, not how it changes when it gets better or worse. Brain impacted by body, very little work actually measuring brain changes associated with bodily illness or medical intervention. And again, environmental factors and how they're the gift that keeps on giving and causing ongoing problems day to day. I started a research program called Transcend, which means treatment research and neuroscience evaluation of neurodevelopmental disorders, and we're trying to do all of this, measuring different parts of the brain in relation to body function to look at change. Change in development, change in regression, and change in improvement. And also, hopefully, we'll have the resources soon to build an archive of clinical EEGs, which our engineers can study. So if you have a baseline EEG and you have either worsening or improvement, hopefully, we can analyze what changes in the brain in some of the measures I showed you. I'm very excited about a study that we've recently launched called a multi-system evaluation of infants at risk for autism. If we're talking about biological problems and whole system problems, we really want to know, when does this start? Are you born with all the abnormal biology in place, or does it develop over the first few years of life? No one has looked. We are, to the best of my knowledge, the first people taking a medical approach to development. There are a lot of people who've taken a behavioral approach, some people who are starting to look at environmental prenatal influences, but not the ongoing approach of changing how does the metabolism, immune system, and brain electrophysiology change. We're looking at EEG that I showed you, sensory responses in the brain, metabolism, lipids, immune, toxics, nutrition, banking samples, stress measures, neuro and motor exams, and we predict that the biological abnormalities may precede behavior abnormalities, and they may have developmental trajectories, and some people don't really understand that. We predict that environmentally sensitive immune and metabolic measures may predict risk, and that these in the future will provide targets for treatment, that when you see somebody's metabolism going south, having problems, you might be able to intervene before the brain cascades into an autism state in its chemistry and function. So I think it's really important for us to be getting these measures and building this out, and I would appreciate any support and thoughts for expanding the study. The last thing I'm going to talk about is there's a whole movement of autism as being treatable and recovery being possible, and if you go online you can find a lot of testimonials of children who've gotten substantially better from treatment. And I think this is really great, but one of the problems is that there is not a lot of good published literature. There isn't any really documenting biomedical approaches that actually lead to this outcome. People are trying to test treatments one at a time, but they're not demonstrating that there was, I once was lost, I now am found. And I think that we really need to do rigorous work on that. This is a paper, Can Children with Autism Recovery? If so, how? A very good psychological treatment. The biomed people should have been there first. We need to look at individuals rigorously, one person at a time. What were they like at baseline? How did they change? We need to develop infrastructure for that. Databases are in development. People, if you don't document that, when you see improvement, people will say, oh, they weren't really autistic in the first place. They would have gotten better by themselves. They were doing so many things at once that it must have been the ABA and the biomed had nothing to do with it. So you need to have good documentation and research strategy. This is an article I recommend, and I can send it to people. It's called Exceptional Disease Courses After the Use of Complementary Alternative Medicine, Selection, Research, Registration, Medical Assessment, and Research in International Perspective. And the thing is that there are in many areas of medicine reports of unusual, astonishing recoveries from things, but they aren't for whole groups of people, and medical research today does group studies. So how do you bridge the gap between evidence-based and experience-based knowledge? And this is an article that lays out frameworks for agreeing on baselines, how you define exceptional, what sorts of evidence you need to start with to document what was wrong, what you need in the medical record, also how you look at cases where things really didn't go well, and how do we set up an international framework for doing this. So I think that if we have these kinds of improvements, we've got to document them, and it's really critical. It has to be documented at a high-quality level. So as I said in the beginning, there's discovery, problem solving, and sleuthing, which are all functions of science. I think by now you should see that there's a big rhetoric issue in terms of the way people frame the models that we've been talking about. I think that really you see my commitment to changing the model and reallocating resources. So in summary, there's a whole body systems model. I think I've shown you that you can garner a lot of evidence to show that it makes sense and that it's plausible. I think further progress depends on framing autism not so much as purely functional, but having a major functional component. So instead of dismissing the idea of change out of hand, because of course we all know that it's hopeless, you actually become interested and you take it seriously. And we need to document early development, change, and recovery meticulously, both to publish it and to learn from what we are doing. So this is, again, this is my last slide, and this is my research program, Transcend Research. And if you go on into the dictionary and you look up the meaning of the word transcend, it means to climb, to surmount, to exist above and independent of, to be transcendent and excel. And I think together we can all transcend the fractiousness, bring together the behavior, the biomedical, the genes, the environment, and the brain, and help everybody now. Thank you.